Buenos días a todos. Bienvenidos al webinar sobre tendencias de la biotecnología de algas para las aplicaciones agrícolas. Para aquellos que no me conozcan, soy Antera Martel, directora científica y curator de la colección del Banco Español de Algas de la Universidad de Las Palmas de Gran Canaria. Esta jornada está organizada por el Banco Español de Algas de la Universidad, entidad gestionada por la Fundación Parque Científico Tecnológico, en colaboración con la Sociedad de Promoción de Gran Canaria en el marco de la actuación del proyecto Interreg MAC REBECA-CCT, la red de colecciones de cultivos para el fomento de la biotecnología azul en la Macaronesia, y también de la plataforma paneuropea MIRRI, que contempla el área estratégica RIA 4 sobre investigación e innovación en el ámbito de la salud y la alimentación, la agroalimentación y el medio ambiente y la energía. Antes de comenzar con la apertura y las ponencias, voy a darles unos datos importantes para el seguimiento de la charla. Agradecer que tengan sus micrófonos y su cámara apagados durante la sesión, a menos que sea su turno para exponer, con el fin de evitar ruidos que impidan la comprensión. A ser posible, los ponentes rogamos se conecten con auriculares por este mismo motivo. En cumplimiento con la normativa de difusión del programa Interreg MAC, disponemos de interpretación de la jornada en inglés y en español. Para escuchar la sesión en estos idiomas, simplemente deben hacer clic en el botón de interpretación ubicado en la barra inferior y seleccionar el canal español, solo el español. Ese canal servirá para poder escuchar la interpretación en ambos idiomas.
To listen to the interpretation in either language. On the toolbar at the bottom of your screen, we have a chat, which shall only be used to report any problem or error which may arise in the sessions, audio or video. For the Q&A session, which will take place at the end of the presentations, you can write your questions in the Q&A section. Only questions included in this section will be answered, and we thus kindly ask you not to include your questions in the chat. Finally, please note that this event is being recorded. Therefore, we kindly ask those of you who do not wish to be recorded to please keep your cameras, microphones turned off during the entire session. Without further ado, we shall now give the floor to today's speakers. It is a pleasure to welcome Dr. Roberti, Dr. Ridini and Francioso from the Department of Agriculture and Food Sciences, University of Bologna, who are actively collaborating with the Spanish Bank of Algae and the REBECA-CCT Project in the evaluation of extracts and bioactive compounds from cyanobacteria, microalgae and seaweeds of the Bank of Algae catalogue for agriculture applications, mainly biopesticides. Today, we have the opportunity to present a scientific work, which is part of the results of the REBECA-CCT project and of several scientific publications by the University of Bologna. Without further ado, I now introduce Roberta Roberti. She's researcher and associate professor, Department of Agriculture and Food Sciences, University of Bologna. Her presentation concerns new solutions to manage plant pathogens by using bioproducts for sustainable agriculture. The floor is over to you.
Yes, I'm here. Thank you, Antera. I'll start to share my presentation just now.
Vai in alto con il mouse, vai in alto con il mouse. Qua, adesso che video, ok.
So, can you see the screen?
Sì, sir.
Okay. Good morning. I'm Roberta. I'm from the Department of Agricultural and Food Sciences of the University of Bologna. I'm an associate professor of plant pathology and plant disease management, working in a team with the two next speakers, Hilary Reghini, she is a research fellow, and Ornella Francioso, which is a professor of chemistry. First, I would like to begin by thanking Antera for inviting us to present our research in this webinar. The research we will show been obtained under an agreement of REBECA project between the University of Las Palmas and my department. So today I will give a talk about new solutions to manage plant pathogens by using bioproducts for sustainable agriculture. So my presentation is divided into two parts. In the first one, I'll give you some information about plants, pathogens, and the need to manage plant pathogens with sustainable means. In the second part, I will show you some results, some representative results from our research. So, talking about plants, surely you all know the importance of plants to life on Earth, because plants are essential. Without them, life as we know, it could not survive. You know that the plants are source of the oxygen we breathe, they capture carbon dioxide, and most important, they provide the base for all food on Earth. And this applies to humans, animals, and microorganisms. So talking about food, I would like to emphasize that plants are sources of many substances. For example, proteins. This is just an example of the importance of plants for our diet by considering the role of proteins. But why proteins? Because they are essential for life. They contain essential nutrients for life, for our life, and have important functions in our bodies. For example, building and repairing tissues, producing enzymes, hormones, and overall maintaining our health. You can see here that proteins can be obtained from various sources, including animal and plant-based foods. Exploring the contributions of these sources to meet the protein need of the world population, this graph indicates that plant-based food sources make substantial contributions by 71% to global protein needs. Moreover, crop production has even a key role in feeding farm animals, 29%. Therefore, we can summarize that altogether proteins from both plants and animals guarantee 97% of the protein need of the world population. You should also know that plants are constantly under pathogen attack. A plant pathogen can be defined as a causal agent of an infectious disease, and we have different plant pathogens attacking plants. Major groups are fungi, bacteria, phytoplasmas, viruses, and nematodes. These agents can cause diseases that are defined as animal functioning of plants, of the growth, reproduction of plants, causing a continuous irritation This results in visible symptoms. In the pictures, you can see some common disease symptoms, root and foot rot. You can see grey mold on grapes, foliar necrosis, symptoms very common both for fungi and also for bacteria, and powdery mildew on cucurbit leaf. So we said before that plants provide the base for all food on Earth, for human, animals, microorganisms, and so on. But the question is, who eat first? Who will be the winner in this competition? About 60 years ago, Horsfall and Diamond stated that since plant pathogens must eat too, they compete with us for our food supply, and they sit down first at the table. Well, these words are very important today, even more important today than about 60 years ago, because the world population is expected to reach an average of 9.7 billion in the 2050. So pathogens, we say that sit down at the table to eat before us, causing crop losses. And if we consider crop losses, statistics show that around 68% of annual worldwide losses are caused by biotic factors. And among biotic factors, there are pathogens that are responsible for 24% of losses, of course. And if we consider fungi, they are the most numerous agents of plant diseases, causing 70 to 80% of the diseases. Therefore, to secure food for the world population, there is a need to reduce crop losses. And to reduce crop losses, the management of plant diseases plays a pivotal role. We have many methods available for the management of plant diseases. And for decades, the main effective method has been the use of chemicals. However, chemicals are associated, the use of chemicals is associated to several problems. We have, you know, that pesticide can contaminate soil, water bodies, leading to the pollution of ecosystems and potentially harming non-target microorganisms or organisms, thus reducing the biodiversity. Moreover, pesticides can also pose risks to human health, both to farmers and consumers. And important to say is that pesticide can persist on crops even after their application. So the residue management is very crucial to minimize the potential risk for health associated with consuming pesticide-contaminated food. And then we can see that pathogens can develop over time resistance against many recent pesticides. So addressing these critical issues requires a shift towards more sustainable approaches to pest and disease control in agriculture. So consequently, the regulation 1107 of the 2009 concerning the placing in the market of pesticide and the directive, the European Directive 128 of the same year concerning the sustainable use of pesticide have encouraged alternative sustainable approaches for the management of plant disease to reduce the impact, the risk of pesticides. Currently, the disease management is directed towards sustainable strategies, achieving the objectives of the farm to fork strategy. That is the core of the European Green Deal. Moreover, one farm to fork objective is to reduce by 50% the use of pesticides in Europe by 2030. So, coming now to our research, after this background of the problem of plant disease management, our goal is both to manage plant diseases and improve plant growth by using products derived from macro and microalgae. With the term microalgae, we intend also cyanobacteria. And these products can be defined alternative methods, alternatives to chemicals, of course. All the macro and microalgae used in our experiments were kindly provided by the Banco Español de Algas, and specifically by Antera. Here, I reported a list of the organisms we used: Anabena 2 cyanobacteria, Anabena minutissima and Arthrospira platensis, 2 red algae, Ania adherens and Hydropuntia cornea, and the brown alga Eclonia maxima. The abbreviations indicates the substances we have extracted from each organisms, and we have tested at different doses against different pathogens. WSP means water-soluble polysaccharides, and PBP means phycobiliproteins. To perform our experiments, we have used a different pathosystem. What is a pathosystem? Is an association between a plant and a pathogen. And a system we have used to test the substances against diseases. Concerning leaf and fruit pathogens, one of the first pathosystem we have used consisted of Podosphaera xanthi on cucurbits, zucchini and cucumber. Podosphaera xanthi is the agent of powdery mildew. Powdery mildew is a disease whose symptoms typically appear as a white or grayish powdery, as you can see in the pictures on leaves. It is an obligate parasite. This means that it can be grown only on plant, only on living plant, and we cannot cultivate in an artificial substrate. Other two pathosystems consisted of Botrytis cinerea on tomato fruit and on strawberry fruit. Botrytis cinerea is a pathogen with a necrotrophic lifestyle. This means that it derives its nutrients by killing and decomposing host tissues, producing a soft rotting. The term gray comes from the color of the fungus on the surface of fruits, as you can see here. Is a very common pathogen. Botrytis attacks over 200 crop hosts all over the world, and is a good test for our aims. In these kinds of pathosystems, pathogens were artificially inoculated on leaf or fruit. And before inoculation, we applied bioproducts, of course, on leaf or fruits. In this slide, you can see a summary of all the combination we have studied for foliar and fruit pathogens, the alga, cyanobacterium, the compounds we have extracted, the plant test, and the disease. And on your right, you can see also a screenshot of 5 published papers. Let's see now more in detail, just some example of the results. Here you can see the effect of Anabena minutissima and aniaderens against the leaf pathogen Podosphaera xanthi. Anabena minutissima, the proteins of Anabena minutissima applied at 5 milligram per milliliter gave a disease reduction by 20% on zucchini, while on cucumber, the disease reduction was 40%. It depends from the plants, of course. We have used this difference in the disease reduction. A significant reduction of disease was also obtained with water-soluble polysaccharides from aniaderens, applied at 5 milligram per milliliter. These substances reduced cucumber disease severity by 60%, and this is a very good result. On tomato, phycobiliproteins extracted from Hydropuntia cornea and Arthrospira platensis reduced gray mold disease caused by Botrytis cinerea. In the picture, you can see the effect of phycobiliproteins from Hydropuntia cornea on tomato fruits in comparison with the control. And in the graph, in the first graph on your left, the results of these different concentrations. The disease reduction, the maximum disease reduction was by 78 from 78 to 43%, with a linear dose response relationship. This is for Hydropuntia cornea. For Arthrospira platensis, phycobiliproteins reduced the disease by 72% without dose-response relationship. This means that all the concentrations reduced the disease in the same way. Now we can see the pathosystems we have used to study the effect of the substances against soil-borne pathogens. In this case, pathogens were artificially inoculated in the soil, since they are pathogens living in the soil, and bioproducts were applied on seed. One pathosystem consisted of Pisum ultimum on tomato seedlings, Piscium ultimum cause root rot and damping off. These symptoms typically manifest as the decay or death of young seedlings. Another pathosystem consisted of Rhizoctonia solani on tomato seedlings. In the circle, yellow circle, you can see the symptom of foot rot. And the last pathosystem used is Fusarium oxysporum on tomato plants. So Fusarium oxysporum is a pathogen different from the other two pathogens because it has a particular behavior in the plant. It attacks roots and enter in the growth in the xylem vessels by blocking the absorptions of water from soil. And as a consequence, the symptom is the plant wilting. Coming to a summary of the results, here's the table with all the combination we have used, the cyanobacteria. Note that in green, I have reported cyanobacteria, in brown, the brown algae, Apronia maxima, and in red, the red algae. On your right, the four published paper. So also in this case, we can see some representative results. Phycobiliproteins from Aminutissima were able, applied to seeds, of course, as I said before, were able to reduce the disease up to 40%. In the picture, you can see on your left the control, the untreated control, infected control, and on your right, the treated plants that grown from treated seeds. And the published paper. Talking about, again, about Rhizoctonia solani, water-soluble polysaccharides from anhydrous reduces the disease by 58%. And in the picture, you can see the effect on plants, the effect on the different doses in comparison with the control, that is a market with C slash plus RS. RS is Rhizoctonia solani, the pathogen. RT means the reference treatment, that is laminarin from Laminaria digitata as a reference control. All the concentrations gave the same disease reduction. Here we have the results of water-soluble polysaccharides from anhydrous against Fusarium wilt. You can see in the picture the effect of the three concentration, as well as in the graph. The highest disease reduction was obtained with the highest concentration, the 1.2 milligram per milliliter applied to tomato seeds. The disease reduction was 35%. And you can see that I have underlined that these results were obtained 40 days after sowing. That means that the treatment was applied at seeds 40 days, so before this result, this assessment. this is a very long term effect. This long-lasting effect against Fusarium oxysporum has been even more pronounced in case with water-soluble polysaccharides from Eclonia maxima. Here you can see the effect of the concentration 0.7 milligram per milliliter, Fox mean Fusarium oxysporum. The disease reduction was more consistent than in the previous case. It was by 58% in case of disease incidence, and the disease index, that means the disease severity, was 54%. And also plant mortality was reduced in a very high way by 70%. Also here the results obtained were after 40 days from the sowing. The sowing is the same time of the treatment. So 40 days away from treatment. This is a very long-lasting effect. Coming to the conclusions, we can say that with these experiments, algae and cyanobacteria are sources of bioproducts. These bioproducts are useful for the management of several plant pathogens. Bioproducts we used consisted of water-soluble polysaccharides and phycobiliproteins. These substances showed in some cases a high bioactivity, reducing symptoms caused by foliar, fruit, and soluble pathogens. Results depend on both pathosystem and also on doses we have applied, and also on the way to application. So we can conclude that these substances that can be considered alternatives to chemical pesticides in both integrated pest management, but also in organic production, where we cannot use synthetic pesticides, but only natural substances. Our research are on progress because we would like to test these substances in more pathosystems, and if it is possible, in large-scale experiments by considering more plants in a big place. Thank you for your attention.
Muchísimas gracias a ti, Roberta.
Thank you very much, Roberta. I think you explained the situation very clearly and also the results, and I think that's very promising results, and we have to see how we will then be able to so have more tests about it, and also develop all these results together with companies. We will see that later with Joaquin. I think it's a very important field for the application and the development of all these results in the future, and I think it's something we're working on, which is fundamental as far as the problems which affect food security all over the world. So thank you for accepting this invitation and for your presentation. And now we are going to continue with the presentation of Hilary Regini, who is collaborating also with the same Department of Agriculture and Food Sciences from the University of Bologna. She's worked with her, collaborated with us on various occasions, cultivating algae and performing different experimental work. And her presentation concerns algae and cyanobacteria prime physiological plant changes by improving plant growth and boosting resistance to pathogens. Thank you very much, Hilary, and welcome to you.
Okay, can you see the presentation? Okay. So, good morning. I'm Hilary Righini. I'm a postdoc here at the Department of Agricultural and Food Sciences of the University of Bologna. My presentation will be focused on priming effects of algae and cyanobacteria on plant physiology and specifically on plant growth and resistance to pathogens. So to start, during their life, plants have to deal with numerous stresses, both abiotics and biotics, that can affect plant health and limit plant production. For abiotic stress, I refer to some environmental conditions such as drought or high temperature, while for biotic, I refer to microorganisms. And specifically during this presentation, I refer to fungi, to phytopathogenic fungi. So plants can modulate several physiological responses in favor of their defense in order to overcome such stresses and return to normal basal metabolisms. Also, treatment with delicitors could represent a sort of stress for the plant, since they could be perceived as a stimuli that induce or cause some changes in plant physiology. These physiological changes include photosynthetic rate, changes in cell wall composition, nutrient translocation, transcription of genes, and since we will talk about pathogens, I refer to defense genes. And these genes can encode for proteins with antifungal activity, production of metabolites, both primary and secondary metabolites. And all these physiological changes occur from a complex metabolic responses called pathways, starting with the perception of a stress, which triggers a cascade of molecular events in plants, ending at various levels by physiological and metabolic responses. Importantly, pathogens can severely limit plant growth in terms of seed germination, seedling emergence, plant length, and root development. And these effects are often connected with the physiological changes previously showed. So coming to algae and cyanobacteria, due to their composition, they have long been used in agriculture to enhance soil fertility and crop productivity. Indeed, they were shown to be highly effective in increasing various plant parameters, for example, vegetative growth and yield, chlorophyll and mineral element content of leaves, seedling height and root development. But the study regarding their effects on the plant physiological changes related to plant defense responses against pathogens are more recent and deserve to be more investigated. And our research is located in this point. So what I will show in this presentation regards the application of phycobilly proteins and polysaccharides, water-soluble polysaccharides, extracted from algae and cyanobacteria on zucchini and cucumber leaf and on tomato seed. Leaf treatment, as you see before, consisted in a spray treatment, while seed were treated by immersion. And our research regarded the study of plant defense responses at different levels. So which kind of plant defense mechanisms are activated, stimulated, or increased by application of algae and cyanobacteria compounds before fungal pathogen infection, in order to understand if plant responses induced by these compounds lead to a plant more responsive and already prepared to counteract pathogen infections. As I said before, plant responses to stresses include changes in transcription of genes, encoding for proteins with antifungal activity, production of metabolites and mostly secondary metabolites with biological activities such as antimicrobial, for example, or metabolites associated with the reinforcement of plant cell wall. This is an important aspect since plant cell wall is the first line of defense against invading pathogens. So it is a sort of physical barrier. And when this barrier is broken by fungal pathogens, for example, the plant immune system is activated. And the result of the interaction between plant and pathogen is the induction of this kind of plant responses. So including transcription of pathogenesis-related genes, PR genes, which encode for pathogenesis-related proteins, PR proteins, with antifungal activity. And the most well-known and studied are chitinase and glucanase, genes that lead to the production of a variety of important secondary metabolites, such as phenols and flavonoids with different biological activities. And for example, in this case, the gene PAL is a key gene of these metabolic pathways called phenylpropanoid pathways. And during our study, we have correlated all this kind of plant responses to the disease control we have observed in our experiments. So as already said, the significance of PR proteins lies in the fact that they show a strong antifungal activity. And in particular, chitinases and glucanases have a role in the degradation of fungal cell wall components, such as chitin and glucans. And their increase in plant is considered a molecular marker of plant-induced resistance against fungal pathogens. Moreover, plants can produce a variety of chemical compounds, secondary metabolites, such as phenols and flavonoids, which are involved in biochemical defense against pathogens and also stress in general. They are known also for participating in plant protection against stresses due to their antimicrobial and antioxidant properties and for their role in the reinforcement of the plant cell wall. But the production of all these compounds and enzymes is regulated at upstream gene levels and follows different plant metabolic pathways. So plant defense responses, as you can understand, are a very complex network of physiological changes. Coming to some of our results, I divided the results firstly in relation to the type of plant responses and then of the type of plant treatment. So starting with the enzymatic activities, we found that application of Anabaena minutissima proteins on zucchini leaves differentially increased chitinase, glucanase, and also peroxidase activity over the days following the treatment, which were linked to Podosphaera xanthi control, that is the agent of powdery mildew, as you've seen before. Unsurprisingly, these enzymatic activities are not all high every day after treatment, since plants modulate their response in relation to the type of stress, the part of the plant affected, and also the energy cost that this response needs. Concerning tomato seed application of phycobiliproteins and polysaccharides, they showed to increase chitinase and glucanase activity in the future seedling and plants. Indeed, these results you are seeing were obtained in seedlings and adult plants after at least 2 weeks until 40 days after treatment, demonstrating a lasting effect on the induction of defense responses of the plants. For example, on the left, proteins from Anabaena minutissima and polysaccharides from Eclonia maxima and Ania adherens caused a very high increase of seedling chitinase activity, but with different behavior related to the applied compounds. For Anabaena minutissima proteins, there was a kind of a dose-response relationship totally different from polysaccharides from the 2 macroalgae. Indeed, polysaccharides from Ania adherens have increased chitinase more than those of Eclonia maxima. On the right, seed application of polysaccharides from Eclonia maxima caused a very high increase of chitinase and glucanase activities in adult plants with or without pathogen infection. In some cases, we found also a no dose-response relationship between the applied compound and the plant response. Indeed, for example, on the left, treatment on tomato seed with different doses of polysaccharides from Ania adherens increased in the same way glucanase, while for chitinase only the highest dose, highlighted with the arrows, lead to an enhancement. In a recent work, so you can see the results on the right, we investigated what happened in the epicotyl and the hypocotyl of seedlings, of tomato seedlings. Since, as I said before, plant responses can occur differently in the divert plant tissues. And in this case, seed treatment with phycobiliproteins from Anabaena minutissima generally caused an increase in enzyme activities in both epicotyl and hypocotyl. But if we look in details, chitinase were enhanced only in the hypocotyls at the highest doses. But glucanase were enhanced both in epicotyl and hypocotyl in the same way. So we have assumed that the presence of both enzymes in hypocotyls, so in roots, caused the control of soil-borne pathogen Rhizoctonia solani that attacks the collar and roots of the plants. So probably plant modulated this kind of response to the site of pathogen attack. Concerning the production of secondary metabolite, here I reported these results showing that seed treatment with phycobiliproteins and polysaccharides also influenced the production of phenol and flavonoids. We found high amounts of phenols in the hypocotyl of the young seedlings that is related to lignin production, and I would to remember that lignin is a phenolic compound present in the cell wall and represent the first line of defense against pathogens. And the young seedlings are more susceptible to pathogen attack, and is very important is presence in root to prevent infection of soil-borne pathogens such as Fusarium oxysporum, which started the infection at root level. About flavonoids, is not strange that they have been found in greater quantities in the epicotyls, so in cotyledons and true leaves of seedlings and plants, since one of their major function is UV protection. At the molecular level, plant response to stress involve modulation of gene transcription. That is what lays behind the production of enzymes such as chitinase and glucanase, and compounds such as phenols and flavonoids. In most of our research, we found that there was mostly a modulation of the transcriptional gene levels, and that not all genes considered are activated or upregulated at the same times. So plants coordinate all this kind of response. In this case, after leaf treatment with Anabena minutissima, we found a modulation of pathogenesis-related genes encoding, for example, for protein with general antifungal activity, the PR1, and for chitinases and glucanases, PR3 and PR2. You can see genes upregulated, highlighted with the arrows, with the blue arrows, at different times after treatment, and the color of the columns indicate the different days after treatment. And in some cases, this upregulation depends on the dose of application. So these results are in line with the enzymatic activities we have seen before, that changes along times and in relation to the dose of the compounds applied. And this is consistent also with the disease control we have obtained, because at each time plant activate or upregulated and produce a response useful against the pathogen over time. Also in this case, polysaccharides from Eclonia maxima applied on tomato seeds increased the expression of genes encoding for proteins with antifungal activity, PR1 and PR3. PR3 are again chitinase, both in presence or absence of the pathogen Fusarium oxysporum. Here also the expression of PAL, the key gene involved in the pathways of phenylpropanoids, was significantly increased, and these pathways lead to the production of phenolic compounds, as I said before, such as lignin, and start other pathways that is related to flavonoids production, in which one of the key enzymes is DFR, that was found highly expressed, as you can see at the right bottom. Also in case of polysaccharides from anidaerens, we found an increase of transcript level of genes encoding for glucanase, PR2, and again of PAL. at any concentration employed in the same way. Moreover, we found high expression of other two genes, HQT, HCT, you can see on the right. And these genes are located downstream PAL, and they regulate the production of chlorogenic acid, that is the most abundant phenolic compound in Solanaceous species, such as tomato. And this phenol has also been reputed to have many important functions in plant against biotic and abiotic extractive. Along with their effect on plant defense responses, we observed also that the treatment with the phycobiliproteins and polysaccharide increased some growth parameters on the same plant. So all these plant physiological changes are connected to each other, since plant more responsive and already prepared to counteract pathogen infections lead to a disease control, and better plant health status lead to a growth promotion. Here, for example, you can see increases of root length and plant height under pathogen infection after the application of polysaccharides from Eclonia maxima. Another case, after phycobiliproteins application on seeds, we found also increases of root length and plant height, along with an increase of plant dry weight, as you can see in the picture on the right. In most of the cases, when polysaccharides and phycobiliproteins were applied as seed treatment, we observed also an increase of seedling emergence over the time, both in presence or absence of soil-borne pathogens. Almost certainly, important metabolic and physiological changes at the pregerminative stage in seed happen due to the treatment and last over the time, influencing the plant defense responses and giving protection to seeds, seedlings, and adult plant, and resulting also in the increases of plant growth parameters. Here, for example, aniadherence polysaccharides increase the plant length and root length in presence of Rhizoctonia solana infection. And in this case, the effect was not related to the polysaccharide doses. Again, aniadherence polysaccharides increase the same growth parameters as we have seen before, root and plant height, in presence of Pythium ultimum. And also here, we did not observe a dose effect. So coming to the conclusions, polysaccharides and phycobiliproteins demonstrated a different physiological effect on plant, depending on their concentration, the pathosystem, and on their mode of application. They were able to elicit several plant defense responses, such as the synthesis of proteins with antifungal activities and of compounds with antimicrobial activity, but also they demonstrated to work at the genes transcription level. All these results showed have been correlated to the reduction of fungal diseases showed in the previous presentation at the beginning. In addition, in addition to elicitate defense responses, polysaccharides and phycobiliproteins stimulated also some plant growth parameters. And taken together, these results make algae and cyanobacteria a source of bioproduct useful for the enhancement of plant defense responses and for the growth of the plants in a sustainable agriculture. And this is all. So thank you.
Thank you very much, Hilary, for your presentation. Also very promising results. We have a few questions, and we are going to answer them at the end of the seminar. And now we're going to continue with Ornella Francioso, who is also a researcher and associate professor, Department of Agriculture and Food Sciences, University of Bologna, and her presentation is entitled Infrared Spectroscopy as an Easy Tool to Characterize Algae, Cyanobacteria, and Plant Structural Changes. Welcome, Ornella. Thank you for accepting the invitation.
Okay. Can you see my slide?
Sí, vemos, pero no vemos la presentación en pantalla completa.
Okay.
Ahora sí.
Thank you.
Perfecto, Ornella.
Okay. Thank you, Antera, for your kind invitation. The topic of this presentation is infrared spectroscopy and easy tools to characterize algae and cyanobacteria and different parts of the plants. And before starting the lecture, it's important to highlight some aspect. What is the infrared spectroscopy? This technique is a very important technique for identifying different functional groups such as methyl, oxidryl, carboxylate, or in organic and inorganic compounds. But it is very important to highlight through these techniques, it's possible to have a fingerprint of each molecule. This is very typical. However, these techniques has the limit when the sample is present in a mixture, meaning the concentration of our compound is less than 1%, and in aqueous solution, because the water absorb very strong in the infrared range. Okay, how does FTR work? This is a very simple introduction. And infrared radiation is absorbed by the bond, in particular covalent and hydrogen bond. And when the motion of vibration of each bond and produce a variation of dipole moment of the molecules. In the picture is indicate the variation of dipole moment. And so when the frequencies of infrared is the same of the vibrational frequencies of the bond, and take place an absorption, and therefore we see a spectrum. Okay. There are two molecular vibrations very important in the spectrum, and are called stretching and bending. Stretching change is regard the variation in the bond length. In the picture, we see the vibration of the water. This is in the stretching symmetric and asymmetric stretch. And bending is the variation in the bond angle, and there are different bending in the molecules, in out plane, in plane. And the particular is the bending take place at lower frequencies. And how is it generated, the infrared spectrum? This is in the slide is represented in a very simple molecules like ethanol.
Ethanol has different covalent bonds like carbon-carbon, carbon-O, carbon hydroxy group, and CH. And we can see the formation of absorption of the energy depending of the type of the covalent bond and the atoms present. And electronegative atoms, for instance, on OH, is absorbed at high frequencies. This is different in CH. This is very important. Two aspects in the generation of the spectrum of the mass of the atoms and the strength of the bond. This influence the vibration. Okay. Why is it important to apply FTR in our research? There are a lot of applications in the literature. For instance, is very, very useful tools in the search of macromolecules. It's possible to investigate the structure of proteins and the carbohydrates and lipids and nucleic acid and also another important application regarding the environmental and agronomic field. For instance, in the decomposition of different parts of the soil, organic matter, humic substances, and the mineral compounds. And the presence also in pollutants, herbicides, pesticides, is possible to have in particular fingerprints of fertilizer, biostimulants, and also microplastics. Another application in the food industry, in the company, to characterize the chemical properties of the food, of forensic company for identification of illegal drugs. Okay, but now for us it's very important the application of infrared to investigate plants and all modification produced by the biotic and abiotic stressor. In the literature, there are a lot of applications. I have reported only the more recent publication. For instance, one important application is the roots and leaves variation associated with the saline stress under long-term treatment, or the modification of the fruit in presence of the infections of viruses. The advantages of these techniques regard first is possible to use small quantity of the sample, raw sample, without pretreatment. Is possible to recognize different macromolecules present in the sample. Okay. It is after the recording the spectrum is important to elaborate, to process the raw data. And the first step is to use baseline corrections, normalization, osmotic methods before to apply other procedure. But it is important to identify the biofingerprint. Is the region is unique for each molecules. Is the range from 1,800 to 900 centimeters minus to -1, and this region can be reprocessed by using correction, normalization, osmotic methods. Okay. In our research, we have applied curve fitting. Curve fitting because we can see a typical spectrum of Anabena water structure. Anabena is very rich in proteins and are indicated in the region, in green region, the presence of protein, amide 1, amide 2, and amide 3. There are a very broad band, and by using curve fitting, in particular, there are a lot of type of algorithms can be used. In this case, the best fitting was used by Gaussian curve fitting. It's possible to observe a different band in this region, in the biofingerprints, and related to methyl groups and CO groups. And the validation of curve fitting take place through the statistical analysis. And I have included some important parameters like reduced chi-square. This value has to be very, very low, and R-square and standard error. These values means there is a good fitting of the experimental data with Gaussian curve. Okay, let's go and our results. In this picture are represented the spectra of Anavena, a water extract from Anavena mintissima, Ectonia maxima, and Jania. And the spectra are completely different. To be more easy, I have included some part related to typical macromolecules present in these compounds, like proteins, alginates, carbohydrates, and carbohydrates in general, lipids, and phosphorus, and carbonates. And Anavena is very rich in proteins, is dominant. This is correspond the band and related to amide 1 and 2. This is the lipids compounds. In Eclonia is dominant the polysaccharide and the sugar and polysaccharide compounds. This is a typical region associated with carbohydrates. And in Jania, instead, is dominant the mineral components like carbonates. This is a typical vibration of carbonates. This is one, this, and this. And when we are investigating the roots, also in this case we have a variation of the relative intensity of some band. The profile is very similar in the different roots. In the black is represented the control, and the other color in red and orange correspond different treatment with water extract, quindi Ania, Anavena, Eclonia, and Jania. And the treatment were done with at different concentration, and orange correspond to 5 milligram per milliliters, and the red, the highest concentration, 10 milligrams per milliliters. And we can see that in Anavena, there is not an effect on the root of the different concentration. But in both treatment, there is an enhancement of the relative intensity which compared with the control. It's different in Eclonia. Eclonia is the highest effect was observed at the concentration at 10 milligrams per milliliters. And this is a result was supported by the previous lecture of Hilary, and with the results obtained by using enzymatic activity. In particular, in Eclonia is increased the component, the aromatic components. In Jania, is the fact is better of the lower concentration as it increase the components related to aromatic rings. Okay. About the components of these the algae, and the microalgae. And we have analyzed the phycobilic proteins in Hydropuntia cornea and Arthrospira platensis. And also in this case, the spectra is completely different, because in Arthrospira platensis is dominant the presence of the proteins. In Hydropuntia cornea is dominant the components related to the carbohydrates. And in more detail, it's possible to have a semi-quantitative of the compounds presence or the functional groups present in the phycobilic proteins. This is Hydropuntia is a very low content in amide 1 and amide 2. Is in the less of 20%. Instead, is increase the components of alcohols and CNC. This is an aliphatic compounds. In Arthrospira platensis, this is very dominant the presence of the amide 1 and amide 2. This is very low the component related to alcohols. And regarding polysaccharides, water soluble, this is was obtained by Eclonia maxima, and the spectrum is typical of polysaccharides. And we have the table is reported the tentative attribution of the main functional groups. This is the region 1,000 from 1,600 correspond to the typical region of polysaccharides. It's possible to identify the type of carbohydrates, for instance, a pyranose ring, or the presence of manuromic acid residues, or the variation of the bending in pyranose rings. Okay. And the effect of the application of infrared on the fruit is very, very, very important. In this case, we have analyzed the tomato fruit and infected with Botrytis cinerea. And in the picture, we can see 3 spectra. In red correspond the control and non-infected. In black correspond the spectra of tomato infected by the pathogens, and in pink correspond in violet in the fruit treated phycobilic proteins. And in this experiment, we have analyzed before the outside skin of the tomato, and it's possible to observe different patterns in each spectrum. In particular, in infected control, the black line, there is decrease the region assigned to polysaccharides, in particular sugar and uronic acid, we have identified, and decrease also the presence of the aromatic ring. In the fruit treated with phycobilic proteins and after infections, we can see a great enhancement of the sugar compounds, and increase also the presence of aromatic rings and phenols, and also polygraptonic acid. Okay, and in the inside of the part of the fruit and the pulp of the tomato, we have analyzed the different parts, and in black correspond the infected, and red correspond to the control, and violet is infected but in presence of phycobilic proteins. And also in the pulp tomato, we can observe an increase of the polysaccharides region, the pectin in particular, and also there is an increase of amide groups. Instead, in the infected, in black, there is a modification of the pectin and increase in the proteins part. And in order to understand in the percentage of each part identified in the spectra, we have performed the curve fitting and reported the histograms of the different percentage area. And we can see that in the infected control is dominant the part related to the protein in blue. And there is a very, very low concentration of pectin region. And instead, in the treated with the phycobiliproteins, the percentage is very similar to the control, but it's very incredible is there is not a modification of the region assigned to the pectin. Okay. In conclusion, and we can conclude that infrared spectroscopy is very, very useful tools in providing valuable information on, in general, functional groups of each sample. But it's possible to identify in chemical structure like polysaccharides and phycobiliproteins from different origin. And in particular, the techniques is very useful to investigate the route and fluid before and after treatment and in presence of the different pathogens, because it's possible to have an idea what is the effect of the pathogens on the plants of different parts of the plants. And it's important to emphasize the increase in the case studies in order to produce a big data set for accurate disease emergency predictions. Thank you for your attention.
Thank you very much, Ornella, for your didactic and very clear presentation of the technique and the results, which are so interesting and that you're obtaining through the application of water-soluble polysaccharides derived from these algae and cyanobacteria. So it's very promising future as well. Now, I'm going to introduce, today we're also joined by Joaquín Pozo Dengra, who is Director, R&D Director and CEO of the Spanish Biotech Group. He's specialized in the development of eco-sustainable and efficient solution for fruit and vegetable production using bacteria, cyanobacteria, and microalgae. I would like to thank Joaquín, with whom we have shared research work, such as several projects, such as Sabana, and he's going to talk about algae-based biopesticides and biostimulants for agriculture, from research to market reliability. Thank you very much, Joaquín. Good morning, and thank you very much for the invitation. I'm going to share my screen.
Ya está en pantalla completa, ¿verdad?
No, todavía no.
A ver. Voy a hacerlo de otra manera.
Ahora sí.
¿Se ve ahora?
Sí, perfecto, ahora sí, Joaquín.
Perfecto, muchas gracias.
Muchas gracias a ti.
Bueno, pues buenos días, sí, como decía Antera, gracias por la presentación. Soy
Good morning, and thank you for the presentation. I'm Joaquín Pozo. I'm R&D Director of Burroughs and Biotech Group. We started as a company at the end of 2010, focusing on the development for agriculture, algae-based products for agriculture. And we based ourselves on previous work and also on a personal level. And the present is seat is in Almeria, and it has different companies also in different countries. You can see here, such as India as well, and the Philippines, and in other countries. We have been the pioneers in the development of microalgae-based products for agriculture based on microalgae. And apart from this initial development, it was AlgaFed, was our first product. Then we continued with biocatalytic processing, with enzymatic process, which we have called TreeTech. And we are just working basically with this system. We are located in the Technological Park in Almeria. We have also contributed to the Sabana project and also the financing. Thanks to this financing, we could build this first microalgae production plant and production with different photobioreactors of a raceway type, and also tubular photobioreactors. The plant is completely automized in a greenhouse, and which we can control in a passive way, depending on the needs, depending on the temperature needed by these microalgae we're working with. Currently, we are finishing a second plant, a facility, production facility. It's on the premises of an electric production company. We work with microalgae, but also with other microorganisms such as bacteria. And since a year, we've been working with different botanical extracts. So why are we working with microalgae? As we've seen in the previous presentations, microalgae are really an excellent raw material when we look for biocontrol. We can find a high protein content, which through different biocatalytic processes, we can transform them in different useful products. And also we can find phytohormones, growth hormones, and plant growth regulatory substances, vitamins, and active peptides, which are generated through the production of proteins. Microalgae have beneficial effects and are very important in plant nutrition and also agricultural production. For instance, we can improve the stress of plants, plant defense against environmental stress and also pests and diseases. We can improve better root development and also better yield. So we are really improving in general the economic yield. If I think about the development of the first product about 15 years ago based on microalgae. We also looked at seaweeds in agriculture. So the person who has worked on this 15 years ago also said, well, if there are products based on seaweeds, why couldn't we develop also products based on microalgae? And what are the advantages and disadvantages between the two? And we are able to develop a homogeneous raw material which can be useful with a high content in proteins and also in phytohormones. And this is how our different products have been developed. Our products have an objective, which is developing sustainable products for agriculture. When we talk about the development of Horizon Biotech, we have to focus on research and development, because we are very active in development and we are obtaining various new products. We can talk about the three research lines, which are the development of biostimulant products, of also prebiotics and probiotics for plant nutrition, and the reducing fossil-based fertilizer. To date, we've developed a large number of projects, both national and international, and I'm going to focus on one European project, which we're currently part of, which is called Algenodes, and whose aim is the development of biopesticides based on microalgae and also based on circular economy to obtain biofertilizers. What we're doing in this Algenode project is to validate the use of microalgae in such a project, and we are transforming biomass in fertilizers through a process which zero residues. These are some examples of the 5 strains we are working with in the same project. So they've been selected based on their biostimulant activity. We can see the results here. We can see auxin-like activity or cytokinin-like activities. And we've also made an evaluation of the germination index when we looked at the dry extracts of these microalgae. We also evaluated the biostimulant activity for this strain. We've seen the root activity, also the leaf activity, and we've compared between the different selected strains, and then an evaluation of field tests we've made in different pots, and we've evaluated the biostimulant activity of the different strains. And now we are in a phase whereby we've selected specific strains, and now we're already working on the commercial part. We can see here, of course, the evaluation of the biopesticidal activity and how some of these strains have shown bactericidal and antifungal activity. And this antifungal is the most important one for this project. Right now, we're working on different field tests. For instance, we can see how after Fusarium infection, we can see that through the treatment with this product developed through this project, we could manage to stop the infection. So we can see the stem right down when compared with the control. Well, we can see the treated one. We can see that we managed to stop the infection by this fungus. So we are still working, developing technology and applications, and right now we're working on a circular economy project, which is called REAL. And the objective is working on waste streams from greenhouses, hydroponic greenhouses, and use this drainage water. You use it in biomass production, and we can then use these products for agriculture or for aquaculture. The project has different validation facilities in Finland and Netherlands, such as Wageningen. And then we have demonstration facilities also, and we're going to build them. So we're going to be able to validate this in Spain and Portugal. And the project is coordinated by Necton, the Portuguese company. This is the consortium in which we are participating. Here are some other projects we're working with. This is with University of Maria and the Cajamar Foundation. So also the aim is really sustainable production, microalgae, and also the creation of products for agriculture. This is really our objective as a company. To date, we've developed a large catalog of solutions so that there are kind of toolkit which the farmer can use and reduce the use of chemical products. So we always continue with our first product, which is AlgaFert in our catalog, which is has active peptides and also many phytohormones. Here we are just doing some tests in Italy for wheat, so we can see the biostimulating effect is clearly visible and also effective in wheat nutrition. We can see how heavier grains are. And also in a vineyard in France, we are comparing right now, and we are carrying out this test where we can see conventional fertilization respect microalgae-based fertilization strategy. So the development of fruit at the beginning, we can already see it's very clearly on these pictures. We've also developed products for better rooting. So here in tomato, the roots and the density is much bigger, especially when we're using algae-based products. And in this case, we have a product which we have developed as a probiotic for rhizosphere microbiota. Is BioBalance, has been developed through the identification of a metabolite, BEA-ULPGC, which is the natural chemical signal which develops this synergic the synergy between the rhizome and the plant. So we have a more fertile soil, and moreover, when we use a combination of this product with a bacteria in agriculture, we're really improving the microbiota. It was just gave you a general overview of the work we're carrying out and the research, which is now ongoing. And now we are already marketing it worldwide. Thank you. Thank you very much for the work you're carrying out, because very important for also agriculture, because of the current food crisis and climate crisis which we're living. And I think that a company like yours, based on agriculture, is very important. So thank you again, Joaquin, for accepting the invitation. Right now, I would like to continue with the Q&A section. We had previously some questions submitted through the registration form, and I would like to thank participants to this seminar. And one of the questions is seen. So what have been the UE regulations regarding use of biomass extracts and their solvents, residues in agriculture? Who wants to answer this question? Maybe Roberta?
No. European Community has established there is not the presence of solvent in the biomasses, in particular if applied to the food. Fortunately, at the moment, there are a lot of new practices used to extract different compounds, biomasses or algae, to obtain proteins, pigments, phenols, and more. And these techniques are supported by fermentation, aerobic and anaerobic fermentation, or it's possible to use a green solvent, and or another type is I don't remember the supercritical extractions. There are not solvent. This is our opinion. I don't know. Joaquin, I think, can improve my response.
If I may just add something, there is a list of residues, of chemical residues, which the EU establishes, and if the compound is not present in this list, then there is no problem.
Depending on the solvent, I think, is the residual, depending of the chemical composition of residues, I think. Okay, no, there is not.
Con el objetivo de usar el agua que es resultante del cosechado del cultivo de microalgas, ¿cuáles son los límites de carbono, cloruro?
And what are the limits on carbonates, chlorides, sodium nitrates, and other substances in the cultivation media resulting from the harvest of microalgae that may affect the growth of plant in agricultural crops and potential filtration into adjacent water bodies?
Okay, if you want, I can.
Okay, Ornella.
Okay. About the carbonates or chlorine and sodium, of course, there are some limitations for applying in agriculture, because high concentration of sodium or carbonates can influence the chemical characteristic of the soil and influence also there are a negative effect on the crops. And for this reason, there are the limits of the use of this effluence of body water derived from the cultivation of algae or microalgae. This is it's important to analyze before use this effluence the content of chlorine, sodium, and for instance, for nitrate, the limits is forbidden the use of nitrates if the concentration is higher and 30 milligrams per liters. This is the and for sodium is the parameters very important is the EC, electroconducibility, and there is a range, and within the concentration of sodium cannot overcome.
So it's possible to consult all these levels.
Yeah, yeah.
As far as spirulina is concerned, is the same question, in fact, because one is the use of spirulina for biofertilizer in the circular economy, and the use of products obtained from spirulina extracts in applications such as biofertilizers, or always in a circular economy perspective. Maybe Joaquin, you can answer this question. The efficacy of spirulina is proved in agriculture, and right now the fact that bacteria cannot be marketed in agriculture. This is the new framework directive from the EU. This has to be based on the register in the EU as a biofertilizer. It's no problem. And this is part of the legislation which is currently in force. Referring to circular economy, it really depends, because what is important is to comply with the requirements for heavy metals and bacteria, and also residues of pesticides. So if in this case, if it complies with this, then the product is can be used without any problem. Concerning algae, which are recognized as also in food also, is there any regulation which has to do with the use? Sorry, it's inaudible. Connection is not good enough. Currently, this only applies. Can you repeat the question, Antera? Yes, I was saying that although there is a list of algae, microalgae, cyanobacteria, seaweeds, and food, there is a list of these algae which can be used in terms of food safety, also, which can also be used in agriculture or for other organisms does it also exist in permitted algae allowed for agriculture? There is a list which, based on the new biostimulant legislation, excludes cyanobacteria. I think there is a mistake because it doesn't make any sense that we can eat spirulina, but we cannot use it in the production of biostimulant. Right now, we're working with DG Agri of the European Union, European Commission, in order to find a solution to this. But according to the current regulation, spirulina as a cyanobacteria, otherwise it can be marketed, but it cannot be registered as a fertilizer, biofertilizer, as others have been marketed till now. Does it have to do with cyanotoxins? It is an interpretation which has been made concerning the production of cyanotoxins by cyanobacteria. But there are dinoflagellates which also produce toxins, are not regulated. So what about that? So who has consulted with whom? This is the question. The fact that there is no general consensus in this new legislation, and this is what has not been done, and now we have to fight for it so that there is a new regulation. So it seems to be quite a common problem within the algae sector. Who do you consult with? And there is this, so do you turn to specialists? The question remains. And as far as aquaponics is concerned, there is a question here. They ask if there are specific products for aquaponics, if there are solutions which can be applied specifically for this sector. Marine aquaponics.
Well, about aquaponics, we have no idea, since we usually not work in aquaponics system. So I don't know. we don't know.
We don't have experience in aquaponic.
But I think that Joaquin can say something about it. Yes, I do have experience. So we're working. What was the connection was not good enough? What was the question exactly? The question was whether these products, which are used for agriculture, soil, can also be used for in aquaponics and marine aquaponics in particular. They are used in case of biostimulants. They can be used in hydroponic systems and also in applications in soil, fertigation, and also used for applications at a foliar application. These products are adapted to any use because the important thing is to provide nutrients that plants are able to absorb them in the best way.
And in my opinion, it's possible to use these effluents, but it's important to control the concentration of chlorine, sodium, and nitrate. This is important.
I think that Joaquin just explained that they have a product which is using this effluent from aquaponics. We have just started to evaluate its use, and the product which we are finding is the high salt content, which is found in this drainage of hydroponic crops, and in Newcastle we had a general meeting of the consortium, and we have discussed how we're going to tackle this high salt concentration and in the cultivation of the different selected microalgae. It has to do with the ability of reducing the salt content. If we have the biomass, there shouldn't be any problem. We could always wash it out, and because the biomass is really independent, doesn't have to do exactly with this directly with this problem of salt content in the medium.
Parece ser que Antera ahora mismo está como desconectada. Vale, espera. Vale, vamos a esperar a ver si se conecta.
Hola.
Hola.
Perdón, es que ha habido un problema, un corte de conexión aquí de la Wi-Fi y me he quedado sin conectar con
Sorry,
ustedes.
there was a problem with Wi-Fi. We didn't have connection, but now it's okay again. So I think that the questions that were asked and submitted through the registration form, we've asked them, and also if there are pigments which may be of interest for agricultural and industrial use. I think that in the presentations of Roberta, Hilary, and Ornella, I think we have had an overview of all these effects on plants. I don't know how this can also be used at industrial level, but as far as the laboratory is concerned, there are already results which have been obtained. I don't know whether anyone of you would like to add anything.
No.
Chiede qualcosa sui volumi, guarda. About,
there is a
question in the chart regarding the volume used, the range of volume needed in milliliter for plant disease management. Do you remember how many milliliter did you apply, Hilary?
Well, at the moment, not so well, but it depends if it is a foliar treatment or yes, or a seed treatment. And for seed treatment, it depends on the amount of seeds I treated. But usually, if I remember well, I think 1 or 2 milliliters for 50 seeds, more or less. For a leaf, I think 1 milliliter for zucchini.
For leaf?
Sì,
for each
for
leaf.
cotyledonary leaf.
Yes.
Nos pregunta por el chat también.
They are asking on the chat also what kind of biosecurity analyses should be made if there is a toxic effect and if this can be found later in the crop.
Toxico para la planta?
The toxic for the plant?
Ah,
for the consumer, for the final consumer.
We don't have information.
No, because first of all, it's important to identify the toxin, and then it's possible to analyze the final product, for instance, the fruit or the leaves, to identify this product. But at the moment, we don't have this information.
We just have an information about the cyanotoxin of the cyanobacteria anabena. Yes, so we have the information about the absence of cyanotoxin of Anabena minutissima. The strain, your strain, Antera.
Sí. También
There are also extracts. In the evaluation of extracts, can toxins be evaluated as well? And then it can be verified on the plant and see the biosecurity. I don't know whether, Joaquin, you've been working on this.
There is two problems. Identify the toxin, it is important to know the type of toxin and the concentration. And because by using infrared spectroscopy, if the concentration is less 1 milligrams, it's very difficult to identify this toxin. It's important to use other techniques like mass spectrometry, massa or mass spectroscopy and other techniques more sensitive, not infrared.
Concentration, toxin concentration, normally is very low at the level which is applied, not only for in our case, because we working with microalgae which are toxin-free, but even if there was a presence of toxins, so per hectare is really minimal. So it would never be found in the final product in case this toxin existed. Let's take, for instance, an example, dry weight, for instance, it could be 5% microalgae in 1 liter, and between 1.5 and 2 liters. We could apply it on 1 hectare. So mixed with another product, of course. So the application percentage of possible toxin is really minimal. Here there is also the director of the biotechnology part of the Spanish Bank of Algae, and would I like to ask, what did it bring to you to be so self-sufficient, this production in microalgae for your company? It was very important for us because facing competition, it was much easier for us because we could produce different microalgae strains and also develop different products based on different algae with different properties. So it's really a very important achievement for companies such as ours. And then it also allows us to have a different product and also generating new strains. So unlimited kind of possibility of introducing new microalgae strains for the development of new products. And as far as the management of the company is concerned, was it easy to combine transformation and production? Was it something natural for you to do that? So the production of biomass is ongoing. We are producing it in the plant, and every day we're harvesting it, and then we just take it to the plant, to the production plant, which is just 15 minutes away from here, and then the products are made. So designing such a plant is rather complex endeavor because there are not many of them, this industrial scale. There are many problems we encounter, and they are different compared with laboratory problems. But when we find a certain stability and we're able to obtain and reach this production level, then it's of course much easier. So the quality of the final product is can be controlled and also improved. So the management of these strains really has to do with the maximization of interesting compounds, these compounds which can be used in products. So it's important to manage different crops, changing and controlling their so we can really work in different fields thanks to all these new developments. And also two more technical questions. So we are now recording this presentation because we will be sending it to you and sharing it with the participants, and also on our webpage and on the REBECA-CCT project and of the Spanish Bank of Algae, and also for these people who couldn't attend. So if more questions concerning all these issues, and also we've received all these questions through the registration form, and if you want, you can also receive a certificate of attendance if you request it. If you have anything else to add, otherwise I will just go to the conclusions.
Just thank you.
Well, I would like to thank again Roberta, Hilary, Ornella, and Joaquin for their availability, their presentations, and for answering the doubts raised by the participants and by our team. And also thank Artemis Rivera, Carla, Christian from the Canary Science and Technology Park Foundation for the technical organization, support and promotion of this event. Also thank our colleague Germana Foscale for the intense interpreting work, and the promotion agency for Gran Canaria for financing the interpretation of this seminar, and to all the partners and colleagues of the REBECA-CCT project. This is one of a series, the last one in a series of webinars. Thank you very much for the opportunity you've given us to show the results and also of learning more about the subject. And also especially would like to thank all the participants for their interest shown and the response given to this REBECA seminar, and which had to do with the trends in algal biotechnology for agriculture applications. I hope we will have a second part in order to have more data on different projects from Biorizon and also the ones we are carrying out, as well as at the University of Bologna. So we will send you the recording of this online seminar, and it will also be available on the REBECA-CCT and the Spanish Bank of Algae websites. And for more information about this project and the activities of the consortium, you can contact us through the information emails provided on both websites. We hope you have enjoyed today's seminars, and thank you, and also we hope to see you again, and sorry for the technical problems we've had at the end of the seminar. So thank you again. So have a lovely weekend, and also
for you.
great holidays.
Bye.